Hello students, today in the biochemistry we are going to start an important topic. This is the amino acid. What is the mean of the amino acid? You already have studied in the previous class that amino acid is the building block of the protein molecule. And we know very much about the importance of the protein in our body. The amino acids is a single unit. Simply we can say that amino acids are the monomers, a number of amino acids bonded with each other by the peptide bond and makes the long polypeptide chain, and ultimately it forms the protein molecule. So today I am going to explain you the basic principle and the chemistry of the amino acids. This amino acids, what is the importance of this amino acid? So first of all, you have to know about the significance of the protein molecules in our body. As we know that all kind of biochemical reactions taking place in our body, which is also called as the metabolism, the metabolic activity is catalyzed by a specific molecule which is called as the enzymes. We all know that enzymes activate the chemical reaction, the biochemical reaction in our body. We know that an essential energy is required for the transformation of a reactant into the product. As we know in the chemical reactions, some of the molecules, just like the A and the B molecules, and reacts and synthesized a molecule C. In such kind of reaction, the enzymes are very crucial. Enzymes are very important for the taking place in this reaction. All the enzymes are made up of the protein. So we can say if there is a scarcity of the protein in our body in the form the enzymes, and ultimately resulting into the decrease of the chemical reactions. So first of all, we can say the all metabolic activities are proceeded by the presence of the enzymes, and enzymes are made of the protein. So here now I am going to explain you about the structure of the amino acids. We all know that the amino acid has two kind of the functional group. I am here writing a typical structure of the amino acid. In all amino acid, a central carbon atom that is called as the alpha carbon, and we know that each carbon atom has the 4 valencies. In every amino acid, first group is the carboxylic group that is called as the COOH, and other functional group which is a important part of the amino acid, that is the amine group. This is the amine group and this is the carboxylic group. And third valency of the carbon is satisfied by the hydrogen, and here I am writing here an R. This R means alkyl group. All amino acids varies to each other by the structure of the alkyl. All the amino acid has a similar typical structure, और जैसा कि इसमें आप दिखाई दे रहा है, but there is a variation in the chemical structure of this alkyl group. Whenever we learn about the different structure of the amino acid, we have to just replace different kinds of the alkyl group in the position of this typical structure of the amino acid. Normally, whenever we study in the biochemical reaction, or you have already studied in the previous classes, that there are 20 kinds of the amino acids for the objective examination. Here I am explaining here about the 22 types of the amino acids. The first amino acid which is discovered that was the asparagine. Asparagine was discovered in the 1806, and the last and the most recently discovered amino acid, this is the threonine, and this is discovered in the 1938. The name of the amino acid, just like the asparagine amino acid, this asparagine amino acid is isolated from the asparagus, and that's why it is called as the asparagine. Similarly, the glutamate amino acid is an important amino acid which is extracted from the wheat gluten, and that's why it is called as the glutamate. The glutamate word is derived from the gluten. And the tyrosine amino acid is extracted from the cheese. In the Greek language, the cheese means the tyros, That's why this amino acid is called as the tyrosine. And here another example, that is the glycine. And that is taken from another Greek word that is called as the glycos, and that's mean in the English is the sweets. That's why its the name is given the glycine. Here, this is a common introduction of the amino acids. As I have already explained here, that any typical amino acid has one the carboxylic group. Here you can clearly seen in the typical structure of amino acid, and this is the carboxylic group, that is COOH. And in this figure, this COOH is represented in the form of, in such like the C double bond O and the OH. This is the carboxylic group. And the other important functional group is the NS2, and this is the amine group. This is the amine group. And other third valency of the carbon is satisfied the hydrogen, and R represent the alkyl group. This is the hydrocarbon group, alkyl group. And one is the important factor, you have to understand that whether the amino acid is present in the dry state or the amino acid is present in the dissolved condition. The amino acids always remains in the charged form, just like the central carbon atom. Here I am writing the alkyl group and hydrogen, and carboxylic group is always remains as the carboxylate ion COO minus by the release of hydrogen plus. This carboxylic group release hydrogen plus, one proton is released, and this proton is accepted by the amine group of amino acid, which in turn convert the NS2 group into the NS3 plus. So we can say the amino acid always remains in the ionic form, whether amino acid is present in the dry state or the dissolved condition. This is the chemical structure of amino acid. And what is the principle of nomenclature of amino acids? First of all, we have to understand the basic component of amino acids. Each amino acids, as we know that amino acid and the proteins are the organic material. So as a basic elements, carbon, hydrogen, oxygen, and nitrogen are the basic elements of any amino acid. But we know that some of the amino acid consisted of the sulfur as a component, just like the cysteine and the methionine. What is the cysteine? Here I am drawing here the what is the alkyl group of the cysteine. This is the cysteine. One is the carboxylic group, other is the amine group and hydrogen group. And on the position of alkyl group in the cysteine, there is CS2SH. This is the thiol group is present as the alkyl group. CS2SH, this is the alkyl group of the cysteine amino acids. This is the cysteine. So we can say the cysteine has one sulfur in the alkyl group. And another example of sulfur containing amino acid is the methionine. Here I am writing here the structure of methionine. This is the alpha carbon is bonded with the carbon, and in place of the alkyl group there is a long hydrocarbon chain CS2S and CS3, and this is the alkyl group of methionine, and one sulfur is present in the side chain of the methionine. So whenever it is asked in the objective examination that which amino acid consisted of the sulfur as a basic component, then we can say the cysteine and the methionine. And the presence of cysteine and methionine makes this amino acid the very important, because such kind of amino acid is able to make the disulfide bonds, which is explained in the further the slides. So this is the basic structure of the amino acid. Whenever a carboxylic group, this carboxylic group is attached with a long carbon chain, Okay. So then the carbon group which is directly attached, directly attached with the main carboxylic group is called as the alpha, beta, gamma, and the epsilon. And I here I am making a structure of the molecule which I am going to explain. Just like one molecule is a CH3, and here a CS2 a long carbon chain, and finally a carboxyl group is attached with a long carbon chain. And here the just near to the functional group, the first carbon is called as the alpha carbon, and next to the alpha carbon is the beta carbon, and next to the beta carbon is the gamma carbon. Similarly, if amine group is attached with the alpha carbon, such kind of amino acid is called as the alpha amino acid. If this amine group is attached with the beta carbon, then this amino acid is called as the beta amino acid. The acid is the principal functional group, while the amine group is a subsidiary functional group. Then the nomenclature of amino acid is based on the position of amine group. If amino group is attached with the alpha carbon, then it is called the alpha amino acid, otherwise beta amino acid or the gamma amino acids. What is the standard or the proteinous amino acid? And as its name implies that it is the proteinous amino acid. And what is the mean of the proteinous amino acid? The amino acids are which participate in the protein synthesis. This kind of amino acid called as the proteinogenic or proteogenic amino acid. This amino acid is the proteogenic amino acids. Any chemical compound which has the one carboxyl group and one amino group is called as the amino acid. But not all the amino acid participate in the protein synthesis. So only that amino acid which are participate into the protein synthesis, like the translation process, then it is such kind of the amino acid called as the proteinogenic or the standard amino acids. So there are a number of amino acids found in our body in the different quantity. These amino acid I am here talking about the leucine, serine, lysine, and the glutamic acids are more abundant in our body and found in the much more amount in the body, and while the tryptophan and the methionine and types of the amino acids are commonly called as the rare amino acid because it is found in the minute amount. And here I am saying about the 21st amino acid, that is called the selenocysteine. Normally in the previous classes in the UG and in the school time, we have learned about the total of 20 types of proteinous amino acid. But here I am going to add 2 more amino acid. The 21st amino acid is the selenocysteine. This is the selenocysteine. And what is the selenocysteine? As its name implies, this is the modified amino acid of the cysteine. And here I am making the structure of this selenocysteine. I have already told that in an amino acid, the alkyl group is the variables in all kind of the amino acid. Normally in the cysteine, and there is a CS2SH group in place of the alkyl group, while in the selenocysteine, there is CH2 and selenium and the hydrogen. Means the sulfur is replaced by the selenium in the selenocysteine. So whenever it is asked what is the difference between the cysteine and the selenocysteine, then we can say in the cysteine the sulfur is present in the alkyl group, while in the selenocysteine the selenium is present in place of the sulfur molecule. This amino acid is specified by the stop codon UGA. And what is the mean of this stop codon? We all know that there is a sequence of nucleotides on the mRNA and which specify a particular amino acid during the process of translation process. So this selenocysteine and is specified by the stop codon. We all know that there are three kinds of the stop codons in the translation process, and that are the UAA, UAG, and UGA. These three codons are called as the terminated codon or the stop codon. This UGA genetic code represent the selenocysteine. It has its own tRNA and the anticodon. We all know that that tRNA molecule has the anticodon sequence, and the mRNA has the codon sequence that is the complementary to each other and very specific to each other. But I am here not explaining much more about the translation process, because it is not essential when I am here describing about the structure of the amino acid. So for the purpose of objective examination, you can learn that selenocysteine amino acid is specified by the stop codon, and its tRNA sequence is the UCA. And it may be asked in the competitive examination the name which one of the enzyme consisted of the selenocysteine as a basic component. So here I am giving the two examples, that is the glutathione peroxidase. This is the glutathione peroxidase and the formate dehydrogenase enzymes. These two enzymes has the selenocysteine and as a basic the component in their chemical structure. Okay, and here one of another amino acid, this is the pyrrolysine. And what is the structure of the pyrrolysine? So we already have understood about the structure of the lysine. Here I am writing here the structure of the lysine. The first is the carboxyl group, other group is the NS2 and hydrogen group, and in the lysine as the alkyl group CS2, 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 and the CS2, and the 4 times. This is the structure of this is the structure of the lysine, the R group. And when we compare this lysine with the pyrrolysine in the structure of the pyrrolysine, and what is the difference? And here I am all again and drawing about the structure of the pyrrolysine, and you can compare the what is the exactly difference. Here is the similar structure, CS2, 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 and CS2, and finally in place of the NH, यहाँ पे just second NH, here a pyrrole ring is attached. So this pyrrole ring is an additional structure in the pyrrolysine. This is the pyrrolysine. The pyrrole group is attached. It is the similar to the lysine amino acid in some bacterial protein. It is found in the bacterial protein. It may be asked in the competitive examination. It is specified by the UAG genetic code. UAG genetic code, and this amino acid incorporated by the translation machinery when during the translation process, whenever one by one amino acid is going to attached by the peptide bond for synthesizing the translation polypeptide chains. So here these are the some of the amino acid on the basis of the chemical structure. The first is the aliphatic categories. Aliphatic means if alkyl group is specified by an aliphatic group, just like if R of the amino acid, just like the glycine, in the glycine R is replaced by the H, and in the alanine R is replaced by the CS3 means methyl group, and R is replaced by the CH3 and hydrocarbon chain, branch hydrocarbon chain, then it is called as the valine. And if R, in place of the R, there is a chain of CH, CS2 and CS3 and CS3, such type of molecule is called as the leucine. And these are the basic structure. if you learn the basic structure of the different kind of the amino acid, then you can compare compare about the structure of different kind of the amino acid. Aromatic amino acid like phenylalanine, tyrosine and tryptophan, and phenylalanine and tyrosine tryptophan has the benzene ring, just like I am here making the structure of phenylalanine. R is replaced by in the phenylalanine CH2, and here is a benzene ring, and this structure is called as the phenylalanine. And if I attach the one hydroxyl group with the phenylalanine ring, then it is called as the tyrosine. And this is the basic difference between the phenylalanine and the tyrosine molecule. If it is asked into the examination, what is the difference between the molecular weight of these two amino acid? Difference between the molecular weight. In the phenylalanine and in the tyrosine, there is a difference in the one hydroxyl group. And we know the atomic weight of hydrogen is the 1, and atomic weight of oxygen is the 16, total is the 17. Then we can say there is a difference between the molecular weight of the phenylalanine tyrosine is the 17. So the basic structure of chemical structure of amino acid should be learned if you want to attend any objective the questions. Here the sulfur containing, I have already explained about the cysteine and the methionine, and I'll draw the different structure of the amino acid again and again and to learn. Then on the basis of chemical structure, the amino acids are classified on the basis of alkyl group. Actually, these amino acids are classified just like the non-polar amino acid. This one number, you can clearly see the non-polar or the hydrophobic amino acid. And what is the mean of this hydrophobic nature? What is the reason of this hydrophobic nature? Just like the alanine amino acid has only methyl group in place of the alkyl group. And similarly, other example is the valine. In the valine, R is replaced by this structure CH, CS and CS3. And this structure is totally hydrocarbon chain, doesn't have any kind of the ionic form. That's why it is the non-polar alkyl group in the valine, and non-polar group in case of the alanine. So these are the non-polar amino acid. They are not able to dissolve in the water. That's why such kind of amino acid is called as the hydrophobic amino acid. And second name is polar or the uncharged amino acid. Means such kind of the amino acid are uncharged but showing the polarity, just like the structure of the serine. In the serine, an R is replaced by in the side chain CS2OH. This is the serine, the side chain of the serine. In the serine, there is no positive or the negative charge in the alkyl group, but due to the presence of this OH group, hydroxyl group, this amino acid showing the polarity. So it is also asked into the examination, write the name of the amino acid and having the uncharged alkyl group but showing the polarity. And these the examples you can see in the second category, the serine, threonine, cysteine, tyrosine and so on. And third example is the positively charged amino acid, just like the lysine. I have already explained about the lysine, and here I can draw the structure of the lysine. And the R group is specified by the CS2, CS2, CS2 and CS2 and NS2. This is the alkyl structure of the lysine. The lysine, in the lysine, this NS2 group is transformed into the NS3 plus in the ionic form. That's why showing the ionic nature and ionic nature, and this ion makes the lysine the polar molecule. These are the positively charged amino acid and showing the basic nature. And another example in the fourth category, you can see the negatively charged amino acid have the side chain, and aspartic acid and the glutamic acid shows the side chain COO, CS2 and COOH, which may be written as CS2COO minus in the ionic form. This is the structure of aspartic acid. This aspartic acid is ionic and the polarity showing the negatively charged and acidic in nature. Similarly, in the glutamic acid, the R group is CS2 2 times and COO minus. This is the glutamic acid. Aspartic acid and glutamic acid both are the negatively charged and show the acidic nature. And whenever you want to learn about the different structure of the amino acids, just learn the structure of the amino acid, then you can attend different kind of the short exam, objective multiple choices examination. This chart is showing the classification of amino acid, and here I am not going to explain. I am talking about the non-standard amino acid. As I have already explained, the amino acid and which doesn't participate in the protein synthesis, such kind of the amino acids are called as the non-standard amino acid. These amino acids are not incorporated in the protein synthesis during the process of translation. Just like these are some of the examples, like the proline. Proline, if a proline is converted into the 4-hydroxyproline, this is the modified amino acids. Similarly, if the lysine is modified into hydroxylysine and lysine in the demosin, the serine in the form N-acetyl serine, azaserine, and the methionine in the form of N-formyl methionine, and the glutamate. These are some of the example. This table is very important and for the objective examination or the short type of the examination. And what is the azaserine? And so you can say the azaserine is the antibiotic, and you can say the don't participate in the protein synthesis, an intermediate product of the metabolism, and another citrulline and the ornithine. You have already studied in the previous classes in the process of synthesis of the urine by the process of ornithine cycle taking place into the liver, and there the citrulline and ornithine is synthesized. These are also non-standard amino acids. So here this is the classification of amino acid on the basis of requirement in our body. The first is the essential amino acids and the non-essential amino acid. And what is the mean? If you see the name, the essential and the non-essential bit, but both amino acids are essential for our body. Then, and why do we say that non-essential amino acid? Actually, the amino acid which is synthesized in our body. These non-essential amino acids, these 10 non-essential amino acids are synthesized in our body. So there is no need to take with the food stuff. That non-essential amino acids are synthesized in our body. That's why there is no need, there is no essentiality to accept to be taken by the food stuff. The essential amino acid cannot be synthesized in our body, I have already written, and the non-essential amino acids can be synthesized in our body. These are some of the examples of the non-polar and the hydrophobic amino acid. I have already explained, and these are the example of the polar and uncharged amino acid. Polarity, if amino acid side chain is the positive or the negatively charged or showing the polarity, if it may be the charged or it may be uncharged. Just like I have already explained as about the aspartic acid and the glutamic acid. In the aspartic acid, the side chain is the CS2CO minus, and in the glutamic acid, side chain is CS2CO minus. This is the aspartic acid and this is the glutamic acid. The both alkyl group is the negatively charged and the polar. But here in the case of the serine, this is the example of the serine. The R group is CS2OH. This is not the charge, but due to the presence of OH group, hydroxyl group, this R group is showing the polarity. Similarly, in the threonine and in case of the cysteine, here also side chain is a CS2SS. By the presence of this thiol group, by the presence of this thiol group, this alkyl group is not the polar but is not charged but the polar in nature. And similarly in the tyrosine, the side chain in the tyrosine alkyl group, the side chain is CH2 benzene ring and the OH. By the presence of hydroxyl group and the presence of this benzene ring, this alkyl group is uncharged but the polar in nature. And here these are the some of the example of the positively charged and the basic amino acid. I have already explained about the lysine and lysine. Lysine is the positively charged amino acid and essential for the growth and the bone development by the collagen production, and it also play a role in the protein synthesis. The structure of the lysine I have already explained. In place of the alkyl group in the lysine there are 4 CS2 groups, and finally it is attached with the amino group. This is the side chain. This alkyl group is the positively charged and the basic in nature. And similarly the arginine is also a versatile amino acid which support the immune system and the wound healing, helps in the wound healing and the nitric oxide production. And histidine is also an important amino acid which is the positively charged amino acid, and it is a precursor of the neurotransmitters, the histamine. We all know that what is the mean of the neurotransmitters, some of the chemicals which helps in the impulse transmission between the two neurons in the synapse. And these are the example of the aspartic and the glutamic acid. I have already explained the R group. R group in the aspartic acid is the CS2CO minus, and in the glutamic acid side chain is CS2 and COO minus. This is the glutamic acid. Both the amino acid has the negatively charged side chain and acidic in nature. So now I am going to explain about the amino acid ionization and the pH. Ionization of the pH, why are we are saying about the ionization nature? We know in the typical structure of amino acid, I again writing here the typical structure of amino acid. One group is the COO minus, another group is the NH3 plus and alkyl group and the hydrogen group. Each amino acid has one carboxylic group at least and one amino group at least, and plus and minus makes the neutralize. It seems like it is a neutral molecule. So, what is the mean of the ionization, and what is the relationship the ionization of amino acid with the pH? Simply we can say if a amino acid accept the proton means the protonation. If the medium, if the pH of the medium is low, then an amino acid has the tendency to accept the proton, to accept the H plus ion. If the pH of the medium high, then this amino acid release the 1 H plus ion. Means amino acid, the acceptance of H plus ion or the release of H plus ion is dependent on the pH of the medium. What is the relationship between the pH and the ionization? I have explained you in the later in the next slide. And the amino acid, I already have said that always remains in the charged condition, just like the CO minus and the NH3 plus. Means single compound, it is a single molecule which has both the charges, positive and the negative. So it is called as the Zwitter ion. This is the Zwitter ion. This word is taken from the German language. That's mean is the hybrid ion, because both and the positive and negative. This amino acid has both the nature. First group, one is the acidic group and other is the basic group. That's why it is called as the amphoteric or the at the neutral condition. So what is the relationship between the ionization and the pH? I am here explaining you. First of all, I am here going to draw a structure of the alanine. Alanine, in the alanine, R group is replaced by the methyl group. This is the structure of alanine, NH3 plus. Now, in the neutral condition, you can clearly see in this structure that this alanine molecule has one CO minus group and NH3 plus. Plus and minus neutralize this molecule. It seems that during the electrolysis, this alanine doesn't move of any electrodes. If I dissolve this alanine into the acidic medium, means when I provide hydrogen plus ion, means in the condition of acidic medium, when this amino acid dissolved into the acidic medium, and we know that acid release the hydrogen plus, this hydrogen plus is accepted by this carboxylic ion. This carboxylic ion accept the H plus ion from acidic medium. Then what changes will be happen in the structure of the alanine? CH3 and the H is the similar, and there is no changes in the NH3 plus. But after accepting the hydrogen ion, this CO minus carboxylic ion is changed into the COH carboxylic group, and now this amino acid is positively charged and move toward the negative electrode during the electrolysis. Similarly, if we dissolve this amino acid in the basic medium, means that medium which contains a hydroxyl group, in that condition this hydroxyl group of the base pull the hydrogen plus from the NH3 group and makes the water molecule H2O. In that condition, this alanine is transformed into the negatively charged amino acid, just like this. This amino acid in the middle condition, you can see the NH2 is found as the amino group NH3. This in the basic medium, NH3 releases the proton, and this proton is accepted by the hydroxyl group and synthesize the water. In that condition, this alanine is now present in the form of the as a negatively charged and move toward the positive electrode. So we can say that the charge of the amino acid is dependent on the pH of the medium. So that point of the pH, that value of the pH in which a amino acid doesn't move either of the any electrode, the positive or the negative electrode, such value of the pH is called the isoelectric point. Here you can see the isoelectric point. What is the isoelectric point of the amino acid? The character of the pH, the value of the pH at which the net electrical charge is 0 is called the isoelectric point or the isoelectric pH, which is designated as the PI. So this is the relationship between the protonation or the deprotonation nature or the ionization nature of the amino acid on the basis of the pH. And here we all know that the protein is a polymer of the amino acid. The amino acids are bonded with the bond that is called as the peptide bond. Here I am drawing a simple structure of the peptide bond. I am here writing a basic structure of amino acid, and this is a common amino acid NH2, and here is the COH carboxyl group, and here this is another amino acid I am writing here, and this is a typical structure of amino acid. Whenever when two amino acid comes to near to each other, then the hydroxyl group of the carboxyl group and the one hydrogen group of the amine group release the water molecule. These molecules release the water molecule, and then this structure is bonded in the form of just like I am writing here, the R and H. And when this carboxyl group release one hydroxyl group OH, then here the CO, and here the amine group, the NH2 release the one the proton which converts NH2 into the NH, the amino group. Such kind of the bond is synthesized between the amino acid. This bond is called as the peptide bond. This is the peptide bond. This is the peptide bond. So we can say the peptide bond is the basic bond which the bonds the monomers of the protein molecule, or simply we can say the amino acids are bonded with the peptide bond by the process of the dehydration and makes the long polypeptide chain. So thank you so much.